Hey guys, so I want to go over protein synthesis with you. This paper, if you want to draw it, you're welcome to. If you need an extra copy, just ask. So this is all about protein synthesis. This is for topic 4. All right, so protein synthesis is literally just how we make protein. That's what synthesis means, to make. How we make protein. Now this is super important. This is something that literally all organisms do because proteins are like traits or characteristics that we have. So the proteins that we make from our DNA actually make us look the way that we do. So the central dogma, this is like the central belief of how protein synthesis works, is DNA to mRNA to protein. And these are literally just steps of how this is made. So DNA to mRNA is called transcription. It's like I'm transcribing DNA into another language of mRNA. And then I am translating the mRNA into protein. So proteins can't really understand the language of DNA. They need it to be made into mRNA first. And then they can use that to make the protein. So I'm going to go over what this looks like in a cell. All right. So the first thing that we need to go over is that every cell has DNA in it. Literally every cell has DNA. Every cell has DNA. So whether you are a plant cell, a bacteria cell, like any sort of prokaryotic, eukaryotic, you have DNA. Okay. This is important because DNA stores your genetic information in the nucleotide sequence. So because every single cell or every single organism has DNA, every single organism also has a nucleotide sequence. It is the sequence of nucleotides, or those nitrogenous bases, that make you unique. Okay, so if you have a different sequence, you're going to get a different protein, you're going to look different. Okay? So, let's start. Inside the nucleus, this is where transcription occurs. So I start with my DNA, and my DNA is transcribed into mRNA. So what this looks like, transcription. Your annotation for transcription should also always be DNA to mRNA. Let's say you have a DNA sequence of A, T, C, G, C. Okay, this is my DNA. I know this is DNA because it has thymine. If I'm transcribing it to mRNA, now my mRNA base pairs are A, U, G, C. So anytime I see an A, I'm going to write a U. Anytime I see a T, I'll write an A, and then this will stay the same. Those still pair with each other. So that is what's happening here. I'm creating an mRNA strand. I know this is transcription because I see DNA and mRNA. Mainly I see DNA. I'm not going to see that in later stages. Okay. Whew. Nucleus is here. So I've just made my mRNA. This mRNA now leaves the nucleus. DNA cannot leave the nucleus. It doesn't leave because it needs to be protected, because it's the most important thing in order of like telling cells what to do. So my mRNA that was just made is allowed to leave the nucleus. It goes into the cytoplasm to a ribosome. This is the site of translating proteins. So this whole process out in the cytoplasm, this is translation. Translation is when I take my mRNA and I make it into protein. Now the way that this works, so you can't see that, the way that this works is that on my mRNA strand, I'm going to start dividing it into 3. So every 3 bases is going to be a codon. Here, I'll do this as an example. Every 3 bases, and that's nitrogenous bases, And this is on mRNA, not DNA, never on DNA. 3 bases on mRNA equals a codon. Every single codon, 1 codon equals 1 amino acid. So what's happening here is that I have a codon on my mRNA. This is my codon. And then there's an anticodon on my tRNA. And then that tRNA is going to go find the amino acid that it codes for that codon. So the way that we figure that out is by using that codon chart. I realize that this is probably like a lot of information for you. I'm sorry. So the way that we figure out what the amino acid sequence is by looking at this codon chart. Every little amino acid that's represented here is by a little dot. So 1 dot equals 1 amino acid. If I string the amino acids together, that's when I get my protein. So more than 1 amino acid equals a protein. So what would this look like? We're going to start with a DNA sequence. And I'm just going to take you through. Let's start with a DNA sequence of A, T, G, C, G, T. Sure. This is DNA. All right. Now, transcription is when I go from DNA to mRNA. So this part is going to be transcription. So that means that I'm going to use my AUGC pairing rules. AUTAGCCGGCTA. This is now my mRNA strand. Now, for translation, I need to go mRNA to protein. So this part, see how the mRNA overlaps? This is going to be translation. The first thing I need to do is divide this into codons. So that's every 3 bases. So 1, 2, 3, this is 1 codon that's going to code for 1 amino acid. When I look at my chart here, all of these names are amino acids. So these bases, you see how there's U's? These are all the bases for mRNA. This does not come from DNA. So what I'm going to do is I'm going to look at my mRNA. So my first base was U. My second, so that means that I'm going to stay in this row. And then my second base was A, so that means I need to stay now in this box. And then my third base is C. So I'm going to follow this over, and that means that my amino acid is tyrosine. And I'm going to abbreviate this TYR. So that's my first amino acid. My second amino acid I'm going to find from here, that's GCA. So this is G, I need to stay in this row. C, I need to stay in this box. And A, I'm going to be in this row. This is alanine. These 2 amino acids together, this is what makes my protein. So I know this video is long and I know this was a lot, but remember, DNA to mRNA to protein, you need to know what molecules are involved in each process, and you need to know where it happens, and you need to know how to use this codon chart. Okay? Let me know if you have any questions.